Alrighty, hello everybody, welcome to lab 2. So in this lab, you are not going to need access to StatCrunch. You need your lab manual and then access to Blackboard, of course, to submit your work. So in this lab, you are going to be doing the work by hand and with a calculator, so no StatCrunch is needed. Okay, in this lab, we're going to be exploring variability, specifically for a measuring tool. So we have different types of variability. There's natural variability, like variability, a variable varies from subject to subject. So if my variable is height, and then I go measure 100 subjects, is every subject going to be the same height or have the same height? No, there's that natural variability inherent to the variable. And then there's also measurement variability. Measurement variability arises when we're taking measurements and we're trying to get a measurement of the same thing. It could be because of how we take the measurements. It could be because of the measuring tool. And so in this one, we're going to compare variability between two different measurement techniques. We're going to explore calculating actual numerical summaries, and then look at different types of variation, how we can calculate variability, how we can assess variability. So that's the main purpose of this lab. And of course, just like other labs, you are encouraged to work with a partner. The process, the data collection will go faster if you work with the person next to you, with a partner, with a friend, or just the person next to you. And then you can also help each other answer the questions. You have to turn in your own work, but you can collaborate and help each other answer the discussion questions and the calculations as well. Okay, so by now in lecture, you have discussed variability. Variability, it's how spread out values are in a data set. Are they close together to the mean, or are they spread out? So if we look at this visual over here, we have three different samples. We have sample A, sample B, and sample C. These are the frequency curves. We can think of these as histograms. So the bars are just missing. We don't have the bars. But the higher the peak, the more frequently observations are seen there. So which sample has the most variability? Which sample varies the most? Well, we just look at the curve that is more extended. It is more spread out. So looking here relatively, sample A has the most variability. That's this blue curve. And then sample C has the least variability, right? That's this orange curve. Because the values are close together for sample C and they're spread apart for sample A. Okay, so visually, that's how we can assess variability. In this lab, we will actually do this numerically. So we'll calculate measures of variability. Okay, so we are pretending that we're baking some ingredient, or we're baking some recipe, we're following some recipe, and we need 1 cup of an ingredient. Well, we have the 1 cup tool, measuring cup, and then we have the 1/3 cup. And so if we need a cup of the ingredient, we can take 1 scoop using the 1 cup tool, and we're going to use beads. So beads are the ingredient that we need. And then if we have a 1/3 cup and we need a cup, we know we have to take 3 scoops. So using these two different measuring tools, we're going to be calculating variability. We're going to use a 1 cup tool and then a 1/3 cup tool. And we're going to be weighing these amounts that we get. So we're going to repeat this process many times. We're going to randomize the order. So we will do this 10 times for both tools. You'll take 10 samples using the 1 cup tool and then 10 samples using the 1/3 cup tool. And you're going to randomize this order so that no bias is present. You may get sloppy after a while. Try to be as exact as possible, but as you're going on, there may be some inherent bias there. Okay, so in table 2.1, you will put whether you are doing the 1 cup or the thirds cup, and then you're going to be measuring the beads on a scale. How many grams is that weight that you get? Ideally, you should be getting the same weight every single time, right? Because you're trying to get a cup. So you should be getting the same weight every single time, but you should see some fluctuation there. It might not be the same every single time. So table 2.1, you'll put whether you do 1 or a third cup. That order is randomized. And then you'll record the weight that you get in that trial for 20 trials total, 10 of each measurement. Table 2.2, you'll just order these measurements. So start with the 1 cup, order it in ascending order, so smallest to largest, and then same for the thirds cup. So that's table 2.2, just kind of sorting the data. Table 2.3, you need to calculate the 5-number summaries for both the 1 variable and the thirds variable. So the 5-number summary, you've talked about that in lecture, and then you will draw the box plots. You'll have 2 box plots for each variable, 1 for each variable. So here are the methods that I just discussed. Randomize the order, get your weights using the cups, pretend like you're baking, right? And you want to get as exact as possible. Place your weights in 2.1, order the weights in 2.2. You're going to do this by hand, right? So you do not need StatCrunch because on an exam, you'll have to calculate these things by hand. Table 2.3, you'll do the 5-number summaries and draw box plots. You'll draw box plots. You can compare the variability by looking at the box plots and see which variable varies more overall. You might have an idea of which variable that is probably going to be based on the measuring process, but you should see that after you do table 2.3 and figure 2.1, which is the box plots. After you draw the box plot, you're going to go to table 2.4 and 2.5. So table 2.4, you're going to calculate the sample mean and the sample standard deviation for the 1's cup, right? Because you had 10 measurements using the 1's cup, so you'll have 10 values, calculate the mean and the standard deviation. Table 2.5, you'll do the same thing, but for the thirds cup, using the thirds cup. So you'll do this twice. The formulas are in the lab manual for you, so you can follow those guides. And then you'll place your results plus the median, so you'll get the median as well, which is the second quartile, so you'll already have that from the 5-number summary. You'll place your results in table 2.6. So table 2.6, this is a summary for everything. And remember, we're comparing variability, so we're going to be looking at standard deviation here. Standard deviation, that's how we look at variability. The mean and the median, these are measures of centrality or central tendency. Standard deviation, this is a way to measure variability. So you'll compare the standard deviation between the 1 variable and the thirds variable. That's the main idea of this lab, comparing variability between these 2 measurement methods. Okay, and then after you complete table 2.6, you'll answer the discussion questions. Remember, all of your work will be submitted on Blackboard. You can work with a partner or a group, but make sure that you submit your work individually. Okay, that is the end of this video.